Welcome back. This is going to be muscular tissue lecture preview. So we'll cover the first 30 slides, roughly a 20 to 30 minute presentation. I'll tell you the highlights, the key points. So let's jump in. So you guys are familiar with muscular tissue a little bit from our tissue lecture whenever we covered skeletal, cardiac, and smooth. If you remember, those were the three different types. Skeletal was voluntary, cardiac and smooth was involuntary. Cardiac was found in the walls of the heart, had those specialized structures called the intercalated discs. Skeletal muscle was going to be striated, and cardiac muscle was going to be striated, but smooth muscle was not going to be striated. So know the three different types of skeletal muscle. Remember which one's voluntary, which one's involuntary. So skeletal muscle tissue is named skeletal muscle because skeletal muscles move bones. And again, skeletal muscle tissue is striated. This is just alternating light and dark bands called striations whenever you see them underneath a microscope. Skeletal muscle tissue works mainly in a voluntary manner. Again, voluntary means conscious. We consciously move our muscle in order to make our bones move. So its activity can be consciously controlled. So most skeletal muscles are also controlled a little bit subconsciously to some extent. For example, the diaphragm alternately contracts and relaxes without really conscious control. So the take-home points from this slide: skeletal muscle is voluntary. It is striated. This is just alternating light and dark bands. And we know that it can be consciously controlled. So cardiac muscle, remember cardiac is going to be involuntary. It is also striated, much like that of skeletal muscle. It's found only in the walls of the heart. The action is involuntary. Again, it's involuntary. We said the cardiac and smooth were both involuntary. We can't really control those. So contraction and relaxation of the heart, we don't consciously control that. It's initiated by the pacemaker in the heart. It's called the SA node, and you'll learn that in Biology 139. So smooth muscle tissue, again, this is going to line the hollow organs such as the intestines. It lacks striations like that of skeletal and cardiac muscle. I apologize. It lacks the striations of skeletal and cardiac muscle. It's involuntary. It's going to be found in hollow internal structures such as blood vessels, airways, and many organs. So take-home points from these first two slides is know the striations, know which ones are striated, know where they're located. Are they voluntary or involuntary? So these are just generalized functions of muscular tissue. A lot of these are going to make sense to you. So I don't really think it's insanely important to memorize these because, again, they're just intuitive. They make sense. So producing body movements, walking and running, stabilizing body positions. If you notice, you'll get muscular. Your muscles will begin to tense up if you try to maintain good posture for so long. We're just used to slumping over, as I'm doing right now in my chair, right? But a lot of that is due to our muscles are just not used to holding our skeleton up in that position. So moving substances within the body, I guess this is one that you really wouldn't think about, but the heart muscle pumps blood, right? So muscle, aside from it just being attached to bone, it also has a function for moving substances within the body and then generating heat. This is what shivering is. So your skeletal muscles are contracting whenever you are shivering, and it's doing that to keep you warm. Okay. So properties of muscular tissue that contribute to homeostasis. And if you remember, the word homeostasis is just this internal balance within the body. So these are the different functions that the skeletal muscle has in order to maintain this homeostasis: excitability, contractility, extensibility, and elasticity. So the excitability portion is just the ability to respond to a stimulus. So if it has a stimulus, it has an input, it will respond to it. Contractility is the ability to contract forcefully when stimulated. So excitability would be an action potential coming down, and an action potential is just a nerve impulse, right? It's a neurochemical message that comes and causes it to be stimulated. Once it has been stimulated, it contracts, right? And it's able to stretch without being damaged. And then it has elasticity, the ability to return to an original length. So, knowing these four different functions, just knowing kind of the definitions of them, excitability, it responds to a stimulus. Contractibility, it can contract. In the same sense, it can contract and go to a point to where it's not damaged. That's called elasticity. And then it's able to return to its original length. Skeletal muscle tissue, again, these are going to be the different components of the tissue itself. I would like for you to know these as well. There's, again, if you know some of these, the prefix is going to make sense to you guys. So fascia. Fascia is just going to be this irregular connective tissue that surrounds muscle. So fascia actually has I can come here and I can show you what fascia looks like. Fascia. So fascia is going to be what surrounds let's see, here you go. This is fascia. Fascia is just going to be this area that surrounds the muscle. It kind of helps the muscle move, and it also kind of provides this extensibility component. If you've ever heard of myofascial release, this is like a massage technique that they will use in order to help relieve muscular tension. They are massaging the fascia. The epimysium, this is going to be the outermost layer of the muscle. It separates about 10 to 100 muscle fibers into bundles called fascicles. So the epimysium separates the muscle fibers into things called fascicles. Perimysium is just going to surround the fascicles. So you've got the epimysium, which is around, separates it into fascicles. The perimysium is around the fascicles. And then the endomysium separates the individual muscle fibers from one another. The tendon is just going to be a cord that attaches a muscle to bone. And the aponeurosis is going to be a broad, flattened tendon. So here you will see the fascicle itself. You'll see the epimysium is around, separated into fascicles. The perimysium will surround the individual fascicles. And then you'll have the endomysium, which is going to separate it into individual muscle fibers. Okay, so the epimysium is again going to divide it into these individual fascicles. The perimysium is going to surround the individual fascicles, and then the endomysium will divide it into muscle fibers. A tendon, again, it's just a cord that attaches to bone, and then the aponeurosis is going to be a broad, flattened tendon. What's important on this side? Somatic motor neuron. I want you to know what somatic motor neurons do. So these are neurons, right? Neuron is just going to be your basic nerve unit, right? Nerves receive stimuli and they send impulses to different, can be either to organs or it can be to skeletal muscle. So we have an impulse that comes into a neuron, and then from there, the neuron is going to send a stimulus to the skeletal muscle. So the neurons that stimulate skeletal muscles to contract are called somatic. Soma means body, so somatic motor neuron. So I want you to know this definition. Somatic motor neurons are neurons that stimulate skeletal muscle to contract. So we say here the axons of somatic motor neurons typically branch many times. And if you guys remember, the axons are going to be what sends the action potential down to the different units. Each muscle fiber is in close contact with one or more capillaries, and this is just important. We know that blood supply is needed in order for things to live, to bring oxygen and nutrients, in order for things to continue to function. So here, what's important on this slide is understanding what hypertrophy is. So the number of skeletal muscle fibers is set before you're born. Muscle growth occurs by a process called hypertrophy. So hypertrophy. This is an enlargement of an existing muscle fiber. An example of this would be going to the gym. When you go to the gym, you stimulate these muscles by lifting weights. Because you lift weights, this is going to cause an enlargement of the existing muscle fibers. So your muscles will grow through a process called hypertrophy. Testosterone and human growth hormone stimulate hypertrophy. This is why you'll see a lot of bodybuilders abuse these substances. They will inject themselves with testosterone or, in some cases, human growth hormone, and it causes an increase in muscle hypertrophy. So it causes the muscle to grow. Satellite cells are cells that are going to retain the capacity to regenerate whenever you have damage to muscle fibers. What's important on this, knowing these definitions, we're getting down to we've talked about the muscle tissue. We've talked about skeletal, cardiac, and smooth. We've talked about the connective tissue that is going to surround the muscle, the epimysium, the perimysium, and the endomysium. Now we're getting down on a subcellular level, and we're talking about the individual muscle fiber. So these are going to be components inside of the muscle fiber itself. So the sarcolemma is going to be the plasma membrane of a muscle cell. So sarcolemma, the plasma membrane of a muscle cell. The transverse T-tubule is going to carry the action potential through the T-tubules. So transverse T-tubule carries an action potential into the plasma membrane. Okay. So the sarcoplasm is the cytoplasm of the muscle fiber. The cytoplasm is going to be rich in a protein called myoglobin. Okay, myoglobin is going to, if you know within blood, there's something called, there's a protein called hemoglobin. So within muscles, myo means muscle, so myoglobin. So it's a protein, it's a red colored protein called myoglobin. Okay, myoglobin releases oxygen when it is needed for ATP production. And you guys remember, ATP is the energy currency of the cell. So far we've covered the sarcolemma, which is going to be the plasma membrane of the muscle cell. Then there's going to be a transverse T-tubule. And the transverse T-tubule, again, is going to carry the action potential into the sarcolemma, which is the plasma membrane. The sarcoplasm is going to be the cytoplasm of the muscle fiber. And again, this is going to be where myoglobin is found. We know that myoglobin is a red colored protein, which is going to bind oxygen molecules. And we know that oxygen will be released whenever ATP production is needed. So some more terms to become familiar with. Myofibrils are going to be thread-like structures which have a contractile function. Okay? Myofibrils are thread-like structures which have a contractile function. The sarcoplasmic reticulum is kind of like the endoplasmic reticulum of the animal cell. Okay, this is going to be membranous sacs which encircle each myofibril. What I want you to know is they store calcium. They store calcium ions. Okay, that's what's important for the sarcoplasmic reticulum, because calcium, as we know, is going to be important for muscle contraction. Okay. So again, the sarcoplasmic reticulum stores calcium ions. The release of calcium triggers muscle contraction. Then you're going to have filaments. Filaments are going to be two. There's going to be a thick and a thin. The thick is going to be the myosin filament, and the thin is going to be the actin filament, and we will discuss those in a little bit more detail. So the sarcomeres are going to be arranged. This is going to be the basic functional unit of a myofibril, and that's going to be important to know. So the sarcomere is the basic functional unit of a myofibril. So we can start out, in order to understand this slide, by understanding that the sarcomere is going to be the basic functional unit of a myofibril. What are myofibrils? Myofibrils are these thread-like structures which have contractile function. Okay, within the myofibril themselves, you have sarcoplasmic reticulum, which is going to store calcium. It releases calcium in order for contraction to occur, and then you are going to have these different filaments. It's going to be called the sliding filament model, which we're going to discuss later on. It's going to consist of a thick and a thin filament. The thick filament is going to be the myosin filament. The thin filament is going to be called the actin filament. So within skeletal muscle tissues, they have these different zones. Okay, these different zones. There's something called the Z disc, the A band, the I band, the H zone, and the M line. And test riders love for you to know the different components of each individual disc or band, right, or zone. So the Z disc is going to be where the thick and the thin filament overlap one another, right? Um, this is going to separate one sarcomere from the next. The A band is going to be the dark middle part of the sarcomere. The I band is where the Z disc is going to pass through the center of each I band. Okay, it's important to know. Okay, it's important to know if there's a test question, it's probably coming from here. The lighter band contains thin filaments, but no thick filaments, right? There are thin filaments within the I band, but there are no thick filaments, okay? The H zone is the center of each A band, which contains thick but no thin filaments. So again, here we see where there is something unique to these. The I band contains only thin filaments, but no thick filaments, okay? The H zone is the center of the A band. We know the A band was the dark middle part of the sarcomere, right? So the H zone is going to be where thick filaments are found, but no thin filaments. And I'll show you guys a picture of this on the next slide. So the M line, this is just like a supporting proteins that hold the thick filaments together in the H zone. So knowing these two would be important, okay? Here is an example of showing you the Z disc. Here you can see where the thick and the thin filaments are overlapping here. If we have a picture of an I band, I'll show you where the thin filaments are only. So here we go. The I band, you can see that there's only thin filaments present within the I band, right? But where the H zone is, the H zone is going to be thick filaments only, okay? The H zone only has thick filaments, but the I band has thin filaments, okay? And this A band is going to be a zone of overlap, right? Muscle proteins, myofibrils are built from three kinds of different proteins: contractile, regulatory, and structural proteins. Contractile proteins generate force during contractions, regulatory proteins switch the contraction process on and off, and structural proteins are going to link the myofibrils to the sarcolemma. I'm not really concerned about this slide. What I am concerned about is these two contractile proteins, myosin and actin. Okay, I'm concerned about these two. This is test question worthy. Myosin is a thick filament. Okay, myosin is a thick filament. Okay, 1, 2, 3, 4, 5, 6. So myosin has more letters in its name, therefore it's thicker than actin. Actin is thin. Actin has less letters. That's just a silly little way I like to remind myself of things. So myosin's function, it functions as a motor protein which can achieve motion. It converts ATP to energy of motion. Again, I told you that in order for contraction to occur, we're going to have the sliding of these filaments across each other. Myosin and actin. Myosin's a thick filament and actin's the thin filament. And whenever we discussed these different areas here, we were talking about actin and myosin, these contractile proteins. So actin is only present within the I band, and within the H zone, the only thing present is actin, or I'm sorry, myosin. Myosin's only present in the H zone, and it's through these overlap of myosin and actin that we have muscular contraction. So actin again is a thin filament. Actin is going to be a site where myosin's head can attach. So this thick filament is going to attach to this thin filament. There's other little proteins here called tropomyosin or troponin. If you guys have ever heard of troponin, an elevated troponin, this is present in skeletal muscle and cardiac muscle. Whenever cardiac muscle becomes injured, troponin leaks out, and this is how we know that we have a cardiac injury, sometimes a heart attack, depending on how high it's elevated. But for our purposes right now, I think it would be important to know that myosin is a thick filament and actin is a thin filament. Um, um, so actin is going to provide a site where myosin head attaches. Okay, tropomyosin and troponin are also part of the thin filament in relaxed muscle. Myosin is blocked from binding to actin. Okay, myosin blocks binding to actin normally. Strands of tropomyosin cover the myosin binding site, and calcium ions binding to troponin move tropomyosin away from the myosin binding sites, which allows muscular contraction to occur. So we'll kind of go through this a little bit, the sliding filament mechanism. So the myosin head is going to attach and walk along the thin filaments to both ends of the sarcomere. So again, the myosin is going to attach to the thin filaments at both ends of the sarcomere, progressively pulling the thin filaments toward the center of the sarcomere. So I want you to imagine like I want you to imagine it just squeezing and pressing towards the center of this sarcomere. As the Z disc become closer together and the sarcomere shortens, it's going to lead to shortening of the entire muscle. Remember, this comes back to the elasticity of the muscle, how it's able to be stretched without becoming injured. These are different structural proteins. Dystrophin is going to link the thin filaments to the sarcolemma, and titin is going to stabilize the position of myosin. You guys not really too worried about this. Here's another picture again of the different zones: the A band, the H zone containing only the thick filaments, the thick myosin filaments, and the I band containing the thin filaments. One thick filament and a myosin molecule here, just showing you the myosin head. It's going to attach to the actin binding sites. So here, the contraction cycle. The onset of contraction begins when the sarcoplasmic reticulum releases calcium into the muscle cell. That's important to know. Okay, contraction is going to occur as the sarcoplasmic reticulum releases calcium into this muscle cell, where they bind to actin, opening the myosin binding sites. So I'd like for you to know these 4 different steps. These are going to be important. I think that you guys should just write these down, practice understanding the different mechanism, the underlying function of contraction. So in order for contraction to begin, we're going to have ATP hydrolysis. This is where a water molecule moves in and hydrolyzes ATP, and this is going to energize the myosin head. Okay. After this, we're going to have the formation of something called cross bridges. This is where the myosin head is going to attach to the actin binding site. Then we have an important thing called the power stroke. During the power stroke, the cross bridge rotates, sliding the filaments. Okay, this is where the sliding filament model comes in. Then eventually we're going to have detachment of myosin from actin. As the next ATP binds to the myosin head detaches from actin. Okay? So once myosin binds to actin and we have the next ATP binding, it will then detach. So the contraction cycle repeats as long as ATP is available and the calcium level is sufficiently high. Continuing cycles applies the force that shortens the sarcomere. I don't know if you guys have ever heard of a process called rigor mortis. Rigor mortis is which there is insufficient ATP available, therefore the muscle stays contracted and becomes stiffened. Okay, this is what happens whenever someone dies. We don't have any ATP. Remember, ATP is the energy currency of the cell. So rigor mortis will then begin to set in, and you have myosin permanently attached to this actin filament. It's unable to detach for relaxation to occur. So again, knowing these 4 steps, I think would be very important for you guys. ATP hydrolysis. After ATP hydrolysis occurs, we now have sufficient energy for myosin to attach to actin, forming the cross bridge. We have the cross bridge formed. Now the power stroke is going to occur. The power stroke is going to be where the filaments slide upon each other. And once we have now we're going to have ATP move into this new myosin binding site to the myosin head. The myosin head will detach from actin, causing a relaxation. Again, we can continue to contract as long as we have sufficient amount of calcium and ATP. So those are going to be the 2 important things that we have to have. Again, we have to have calcium release from the sarcoplasmic reticulum in order to jumpstart this whole process. So this is a relaxed muscle, as you can see here, maximally contracted muscle. The Z disc has moved closer together towards the concentric line. This is a relaxed muscle right here. You can see how the muscle is stretching out here with 2 sarcomeres, and then you can see how it's shortened. These slides right here, you can view if you're in PowerPoint mode. Unfortunately, I will be unable to show you guys this from this view as I'm sharing. So the myosin head, this is, I guess, a version that it will show you. The myosin head Hydrolyzes ATP, become reoriented and energized. So ATP hydrolysis, okay, it's going to form ADP. Myosin is able to bind to actin to form this cross bridge. Awesome. Once we have the cross bridge, we can then go on to form the power stroke. Okay, the power stroke allows us the thick and thin filament to slide upon each other, and as the myosin head binds ATP, the cross bridge detaches. Okay, that's the final step for relaxation. ATP needs to be present for muscle relaxation to occur. I believe this is going to be where I will stop for the first portion of the lecture preview. I greatly appreciate you guys for coming and learning a little bit before class. Thank you all so much.